Welcome to Bison Jam, the podcast that covers wide-ranging topics from Yellowstone National Park and the greater Yellowstone ecosystem. I'm your host, Hugo Sindelar, an assistant professor of film at Montana State University. Today we are continuing our series about the Hot Side of Yellowstone, where we explore the thermal features in the park. I am joined today by Dr. Bill Inskeep, Professor Emeritus in the Department of Land Resources and Environmental Sciences here at Montana State University. In this episode, we are diving into thermophiles. Welcome to the podcast, Bill.
Thanks a lot, Hugo. Wonderful to be here. I've worked with Hugo folks out there listening sometime in the past, and Hugo got a great sense for the right questions in thermophile biology, so I'm excited to join today. And by the way, Bison Jam is an appropriate name because most of the time you're down at the park, you get stuck in one.
Thanks, Bill. Yeah, so let's just jump right in, Bill. Let's start with what the heck is a thermophile?
Good question. We need to start there, and we can break it down if we look at the word thermophile. And of course, phile it means to love. So this is an organism, any kind of organism that loves high temperature. Now, we need to spend a little bit clarifying what we mean by high temperature. Depending on what type of organism you are, whether you're in the domain eukarya or bacteria or archaea, you might have a different temperature limit, and you might have then a different temperature value or threshold that you would be used to call something a thermophile. So, for example, bacteria and archaea, you have to like to live at temperatures above 60 centigrade or about 150 Fahrenheit, somewhere in that neighborhood. You have to not just have mechanisms of resistance. That's not the point. The point is that your body enzymes and your amino acids are set up to love that temperature. Thermophile. People forget this. People think that these organisms are somehow withstanding the hot water or the high temperature, but that's not the case. They're built to live there. So now, for a eukarya, for like an algae or a fungi, the temperature limit to be known as a thermophile is quite a bit lower, down in the neighborhood of 55 centigrade or maybe 120 Fahrenheit. So the answer can vary based on what type of organism you are, but it's still an organism that loves high temperature.
And let's flip that on their head. Can they live at lower temperatures then, or do they actually need those high temperatures to survive?
They need the high temperature to survive. They're not a thermophile that is adapted to high temperature will not survive even in lukewarm environments sometimes, or cooler.
And you hinted at something there. You kind of explained how thermophiles can come from different domains. Can you to people listening out there, they might not know what a domain is.
Yeah. A domain is the largest division of life forms that we recognize. And so domain eukarya contains all the way from algae to humans. And that's usually multicellular organisms, and with differentiation of tissues. And we go into the domain bacteria. It's a group of single-celled organisms.
Okay.
And then likewise, another domain, which many people might think is pretty similar to bacteria, and they are single-celled organisms, but different enough from the bacteria to be known as a different domain, archaea. So there's 3 domains that cover all of the life forms on our planet as we know them.
And there are thermophiles in each of those domains.
In each of those domains, there's thermophiles. But like I said, the temperature can vary a little bit, of course. And the eukarya would be considered thermophiles at quite a bit lower temperature. The algae, for example, would be considered thermophilic at about 45 to 50 centigrade. So quite a bit lower temperature because they just don't handle the high temperature like the single-celled organisms do.
Right. And so if people are going around Yellowstone and want to see thermophiles from these different domains, and I'm saying see in quotation marks, right, for people listening, like where would you find thermophiles in the park?
We have a cheater, essentially. We can just look for a thermal area, look for a fumarole or steam, or look for hot water. Now, how do you know water's hot when you're not supposed to touch it? And you better be careful, because a lot of people get into trouble trying to touch the water. Not trouble from the authorities, but trouble from falling in and maybe injuring yourself. So you have to be extremely careful. It's better to have one of these tools that can actually shoot a infrared beam at the water surface and return a temperature value, and those are quite handy. Same kind of thermometers that are used when you do your radiant floor heat, and you point it at the floor and you look at the tubes that have the hot water. If you have one of those and you use it in Yellowstone, you can learn a lot then without ever touching the water or risking yourself getting close to the water. But once you know the temperature, then you're going to know a lot about whether or not to expect thermophiles there, because you're going to have to be above body temperature, like our body temperature at 98 Fahrenheit or 37 centigrade. You're going to have to be above that to be considered even close to thermophilic. So we don't want to look in what might look to be geothermal, but we stick our finger in there and by golly, it's cold.
Right.
Which there are such habitats where it might look like a sulfur vent or an iron vent. And we can tell it's geothermal, but it may not be high temperature. And you wouldn't know unless you had one of these nice little gadgets that will return you the temperature if you're close enough.
So we talked a little bit in preparation of this podcast, and you kind of told me there are two important environmental variables that really help determine where different species of thermophiles live. Can you talk a little bit about that?
Two of them, certainly the most important variables, which determine who is there. And this goes all the way from whether or not it might be thermophilic algae to bacteria or archaea. But first and foremost, temperature. That might be obvious to everybody, and it really should be, because it's intuitive. We're talking thermophiles. Well, first of all, you have to be over a certain temperature, like we were chatting. The range varies, but you're going to have to be at least over 50 degrees centigrade to be considered thermophilic, that you actually have adapted to a high temperature. Now, as we increase the temperature from there, and we start going up towards boiling, that's as hot as we're going to get.
Okay.
And keep in mind, in Yellowstone, that's a little cooler than 212 Fahrenheit or 100 degrees centigrade. It's a little cooler because of the high elevation and the fact that the total pressure is lower. So the boiling point is only about 91 centigrade. Actually, 8 degrees, 9 degrees cooler than sea level. Cooler.
And why is that at elevation?
That's high pressure. That's the impact of pressure.
Okay.
At boiling.
It's elevation.
So it lowers the boiling point. That's why when you're in the mountains and you're cooking corn and it doesn't seem quite done, it's because the boiling temperature is lower. You add salt to overcome that.
That's why when I backpack, I struggle sometimes to get my my freeze-dried meals to the right.
That's right.
Temperature.
No, and so the point being, it's good to know the range of temperatures. That's what I'm trying to make this point, because effectively we're talking about a temperature range of about 50 centigrade to 90 centigrade. So that's a range of 40 degrees centigrade. That is a lot of temperature range that we still have to work with, and that will still influence which organisms are there, which life forms are there. And so we've figured out that, yes, there are some thermophiles that like it, let's say, from 50 to 60, some that like it 60 to 70, and you get the idea, and some that like it right up at 90, right up at boiling.
How specific do the species get? Is it always roughly like 10 degrees C different, or some only operate in like a small range? I'm just curious.
That That's a very, very good question in microbiology, just in general, is what is the temperature optima of organisms and how much does it range? Generally, 10 degrees is not a bad guess, it's an educated guess, not a bad, let's say, range to choose. So in other words, a thermophile might be living at 70 degrees optimally, but it probably could survive from 60 to 80. It may not be optimum, and I'm not trying to say who knows. It might be almost dead at 60 and almost dead at 80, but you get the idea. There's a range in temperature.
And what's the other variable? Obviously, temperature is number one.
Temperature number one. Number two, pH.
So give us a little overview. I'm sure
Let's
most
talk
people
about
know what pH. Yeah.
pH is one of those funny things that really everybody's exposed to pH, but I think if you ask 10 people, you'll probably get 10 different descriptions in some regards, but that's why I'm going to start off really simply and hopefully correct, because I was trained as a chemist, so if I get this wrong, then you know that you shouldn't be listening anymore to the podcast. Now, what we have with pH is it's a log, first of all, a log scale of the hydrogen ion activity. So hydrogen ions, what's that? That's an acid chemical species, acidic chemical species. Okay, so we call them protons, we call them hydrogen ions, but this is a measure then. The pH scale is a measure of how many hydrogen ions are in the liquid or in the solution. So it's a log scale, and it's a little bit funny. It ranges from 0 to 14. That's important to know.
Yep.
pH 0 is the most acidic condition on this scale, as we're simply defining it right now. pH 14 is the lowest hydrogen ion. So the bigger number is actually lower hydrogen ion.
Right.
pH 7, I think a lot of people can relate and have heard that that's neutral. pH 7 is neutral.
Right.
So it's not going to rot your teeth out, so to speak. But you put battery acid in your teeth, and that's not going to be pleasant, because battery acid pH 1 to 2 will start eroding the calcium phosphates in your teeth.
Wow.
And so there are actually environments in Yellowstone that get that low of a pH value. So the acidity is extremely high. This makes it difficult for organisms to live and get other nutrients across their cell membranes. They're having to deal with extremely high hydrogen ion concentrations. So they have a different complexion of amino acids in their membranes, and they grow optimally at these low pHs and will not be found. We call them acidophiles. So that would be in a thermoacidophile.
Okay.
We just add on whatever we want to add on, I guess, to qualify what kind of a organism we might have here. This would be an organism that likes high temperature and likes acid. And we've studied quite a few of those. And in Norris Geyser Basin, for example, that's a great place to see low pH environments, high sulfur, high iron. We'll talk about sulfur and iron soon here in the podcast.
And so do we know what adaptations do those thermoacidophiles have that helps them live? Do we know? In that low pH environments versus us, right? You put your hand in there, you're in big trouble.
You know, the low pH of 3 and even 2 and 1, now it gets worse. If you get down to pH 1, then you don't want to be sticking your finger in there. pH 3, though, is like the pH of acetic acid.
Okay.
So, or vinegar. And that's not too awful bad. Your hands can handle that for quite some time. You wouldn't want to soak your hand in vinegar overnight. You could imagine what that looked like in the morning.
Yeah.
I mean, a fixed hand, maybe, perhaps. But so this low pH, pH 3, the organisms, yes, we do know the adaptations. There's a variety. First of all, cell walls have to be adapted to this high hydrogen level.
Okay.
And that comes, generally speaking, from the different types of amino acids that can be used to make a cell wall. Bacteria and archaea have different versions of a cell wall. So they have different ways to actually become and have adapted to be acidotolerant in the first place because their cell wall compositions are a little different.
Okay.
Archaea generally do better under, or I think the more appropriate way to put that is, it's more common to see archaea adapted to the really low pH environments.
Okay.
Like pH 1 to 2. There's some archaea that can handle those conditions. And you combine high temperature and high acidity, and that starts to make it very difficult for anyone to survive, any organism to survive, except archaea. Archaea, they've adapted the right conditions, and cell wall primarily, so that they can still maintain homeostasis between inside of the cell and outside in the external environment. And but archaea are quite adapted. If you go to a low pH, high temperature environment in Yellowstone, it's very common to find things like Sulfolobales, for example. And even Solfatera, it's sort of related to the name of the organism, Sulfolobales, like high sulfur, very low pH, and high temperature.
And are there organisms that exist on the other end of the pH scale, up near like 12 to 14? Do Yellowstone ecosystems encompass the whole pH range?
Yes, it's if we looked at a plot of the distribution of pHs, for example, of all the thermal areas in the park, it would look a little what we call bimodal, or there'd be two peaks.
Okay.
There'd be a peak down in the lower pH. We've been talking about the lower pH environments. And then there'd be a peak that's above 7, up near the 7 to 8 range.
Okay.
And so there's a lot of environments, indeed, that have what we call alkaline environment, or so we might call it a thermoalkalophilic.
Okay.
Or a thermoalkalophile. So an organism that likes high temperature, alkaline conditions. And yes, we have those. This is where bacteria tend to be, they're able to handle this higher pH. And so we get some spore formers, many bacilli. Some of these have been used in the energy industry to try to look for high temperature enzymes and things like that. So we can talk more about that as well. Some bacteria have been especially utilized to try to look for enzymes that are stable at high temperatures and high pH. Because a lot of the conditions in industry might favor, like when you're doing chemical biosynthesis, might favor the high pH conditions. So if you have an organism that lives under those conditions, Yellowstone has been a source of those kinds of thermophiles.
That's kind of talking about pH. Another thing, when we met before this podcast, you were talking about is kind of, there's a big myth about these organisms needing oxygen, right? And can you kind of talk about how they fuel themselves, or what they use for energy, right?
And this is where we want to get in and remind ourselves that we're talking about two major groups of organisms. I don't mean taxonomic groups like the domain bacteria, archaea. Now I mean two major groups physiologically, like specifically, are we a phototroph and we're using energy from the sun? Or are we a chemotroph and we're using energy from chemicals? So notice the words phototroph, troph meaning eat or food.
Okay.
And so phototroph, eat sun, or chemotroph, eat chemical.
Okay.
So where the organisms get their energy is from a chemical species that has extra electrons, like we would call that a reduced species. For example, reduced sulfur. Everybody smelled sulfide.
Right.
When you go up to the hot springs, and a lot of them that have enough sulfide that's coming off in the atmosphere, you can smell it. And you'll know that organisms potentially could be using that source of energy because it's a reduced form of sulfur. It has extra electrons on it. All the organism needs is an enzyme that can convert those electrons to cellular energy. And that's what we have is organisms that can take the energy from either chemicals like sulfur, reduced iron, hydrogen, methane. As long as we have one of those constituents there, it's possible that the organisms could be oxidizing that with oxygen. And this was your question about, well, how do they put the battery together? As we know, to get energy out of an electron, we have to have a battery.
Okay.
So at one end of the battery for a microbe is sulfur, iron, methane, hydrogen. At the other end of the battery is either oxygen, nitrate, or some other what we call electron acceptor. That becomes more difficult. What we've learned is that many thermophiles have oxygen electron acceptors. Some that are specialized to really low levels of oxygen, because it was first thought that most thermal high temperature streams and pools didn't have any oxygen, so that all the organisms there would have been using something else. But as we learn more and more about the low levels required to sustain life, it turns out that many of the thermophiles do use oxygen. It doesn't mean they're not using other electron acceptors as well. The key difference of what I'm trying to say is these are not oxygen-avoiding organisms. These are not anaerobes.
Okay.
No, the majority, actually, as it turns out, need oxygen just at low levels. And there are additional organisms present that are utilizing other electron acceptors. And we can go into that, like sulfate or nitrate. There's organisms that can actually utilize those species as electron acceptors, and that completes the battery.
Where do the organisms get these electron acceptors? Where do they come from?
The things like sulfate and nitrate, and even oxygen. Of course, we know oxygen's in the atmosphere, but the other species are in the water. So a lot of geothermal springs will have nitrate and sulfate, just like a regular stream out in the in the wild that's not thermophilic. So these are natural substances that are not unusual, and other environments that are non-thermophilic would have them as well. So microbes can take a nitrate molecule and convert it to nitrogen gas, N2, or they can take sulfate and reconvert it back to sulfide, which is kind of an interesting. You might think that some organisms could use sulfide and then make sulfide. Yeah. Actually, some have mechanisms to do both, whether or not they're doing such a silly thing. But the point is electron cycling is where the energy comes from with chemotrophs, and you need the battery concept to do that. Okay. And that's a very good way to remember it, and that should lead us to thinking, oh, okay, well, we have different chemicals that can support chemotrophs.
And they're also thermophilic phototrophs, correct? Yes. And so they're just using the energy from the sun?
And in order to do that, they got to have pigments.
Okay.
Capture the sunlight. And of course, that's just like a plant that can do photosynthesis. Now, you might say, well, there's a relationship between plants, the photosynthetic apparatus of a plant, and the bacteria that are phototrophs. And the thermophilic phototrophs are nothing that unique. There's, in and of itself, the photosynthetic apparatus is the same. It's not like there's different evolution or what have you of the photosynthetic apparatus. The key thing to be thinking about with photosynthesis, though, is the complement of pigments that a cell may have to harvest light. And once you harvest light, you can then extract the electrons that are contained, or the energy that's contained in the actual light photon. But first, you have to have a chemical that can be excited by light. And that, chlorophyll, of course, we all know that name, chlorophyll. Now, there's a whole family of other pigments that can be like chlorophyll A, chlorophyll B. Then there are other pigments like anthocyanins. And the reason the pigments are important is they change the color of the organism. So then in the spring, the organism could look green, or could be green, or brown, or orange, or pink, or purple based on the pigments that the organisms are utilizing to harvest light. So all of a sudden, color in the spectrum of a phototroph becomes various possible things from green to orange to pinks to purples. And now we have to take that color, and we had a separate way to look at color of chemotrophs.
Yeah, so let's jump in the color in Yellowstone, right? That's probably, we're talking about, organisms that are at the cell scale, right? And so most people actually, when they see them in Yellowstone, they're experiencing them at kind of ecosystem scale where they're taking over areas around thermal features. And Yellowstone is known for its color around thermal features. So I'm curious, maybe using a few examples from Yellowstone, how color is indicative of who lives there in the thermophile world.
It's a great tool. You can use color extremely well if you can measure both temperature and pH.
Okay.
Now, it would be nice to measure a couple of other things, excuse me, but let's just leave it for temperature and pH right now. There's one very important principle with phototrophs, and this will lead to color, but if you're over a temperature of 74 centigrade, you cannot do photosynthesis because all of the enzymatic machinery falls apart. It just does not handle it. There is no organism to this day that we know about. Maybe scientists are pushing the high upper temperature limit. I don't, have an update on that at the moment. But generally speaking, 74 centigrade is the upper limit for photosynthesis. Okay. So if you're in Yellowstone and you measure a spring temperature of 80 degrees and say, there can't be any photosynthesis there. There can't be any phototrophs there unless they died and got washed in. Well, that's possible. Just like a tree could have gotten washed in. You say, hey, that tree's growing in the thermal area. No, it just fell over in the thermal area. That's different. Okay. And that is, as funny as that may sound, it's very important actually. Because a lot of people will see things in thermal areas and they'll think they're growing in there. Like the litter didn't get there based on it growing there. Right. So you can just think of it that way. There are things that fall into thermal areas. And but if that temperature is higher than 74, there is no way any color that you see is coming from a phototroph. That's how powerful that one measurement is. Now, if it's less than 74, then it's like, oh, okay, now I'm really in real trouble because now I have to consider the color of all the pigments and I have to consider the color of possible chemical processes. So let's give a couple of examples of where these colors can overlap and where it could be confusing.
Okay.
Take Grand Prismatic.
Yep.
That's right in Midway Geyser
Yep.
Basin. It's the one you see pictures of, I think. Everybody's seen a picture of Grand Prismatic? I think so, probably.
And also kind of famous for its color gradations.
And if you say, well, I wonder what the color is in that. Chemical or phototroph? Well, if you look closely at a picture of Grand Prismatic, you'll notice that in the center, there isn't any color except the water, which is a little bluish from maybe the dissolved silica, but really you can tell that there's just water there.
Right.
Then as you go out from that toward the shore, and it's circular, so it radiates, around the circle, and that's what makes it so beautiful from the air. But if we could have a thermometer there, it would tell us a lot. Because what it's going to tell us is that the temperature starts to drop below 74. And it's going to tell us then that the color it's possible that the color is due to pigments, which it is in this case. But if that temperature were greater than 74, there's no way we would ever be able to conclude that it was possibly a gradation of pigments. So what we see there is bands of color. And I don't remember the order. But I think there's some...
Usually yellow, right?
Usually some yellow, usually some green, and some salmon.
Right, salmon, yeah, orange.
And those are then different temperature zones that are perfect for different phototrophs. So it's the perfect temperature for one species of phototroph, and then another species, and probably about 3 to 4 different species, but that live in that gradation. And researchers have shown that even the same species can have a temperature variation in temperature optimum, like Synechococcus.
Okay.
So the same organism can live, as we said earlier, over a 10-degree temperature range pretty easily. But the color is coming from different pigments and or bleaching of different pigments. Chlorophyll, for example, can get bleached, and the organisms can get bleached, and it can look rather pale green as opposed to really bright green if you're shaded. So there's a lot of other factors here, and I'm trying to simplify the issue of just pigments. The variation in pigments is being a source of color change. And of course, even with a given pigment, that can change colors too, a little.
Right.
If you go into a bridge in the winter, that's a perfect example of what I'm trying to get across here. And if you have a green phototroph that in the summer might have been a little faded,
okay,
in the winter with lower sun, all of a sudden it's going to be super dark green and lush, and it's trying to capture more sun because there's less of it.
Right.
So that variation is there, and it's good to know about. But that's a fine-tuned variation as opposed to different pigments, yellow versus brown versus green. Now we go over to the chemistry side, and we have to deal with different colors there.
Yeah,
like it or
let's
not.
dive in.
And that's what's the fun part, is because let's take iron.
Okay.
When it's reduced, it's might be form minerals that are black, like pyrite, black, or iron sulfides can generally be blackish, black, dark color. When you form an oxidized iron, it's going to be red or orange. So if you have organisms that are converting iron from Fe2, the reduced form, to Fe3, which is the oxidized form, they're only getting 1 electron out of that. So they got to spin the bicycle sprocket a lot to get enough energy. It'd be like a hamster on a really small wheel. And that's the reality of it. I mean, we'll talk about sulfur in just a minute, and those guys are running a bigger wheel, if you will.
Yeah.
Because they can get 8 electrons out of every unit of S2- sulfide going all the way to sulfate, which is S plus 6. So that's 8 electrons. If you go iron 2 to iron 3, it's 1 electron. So you got to be spinning the cycle a lot, which means it has a practical side. It means you're going to form a lot of iron oxide. And you're going to see this stuff
on
all over.
Yeah.
If you're in an iron depositing spring. And sure enough, you go to Norris Geyser Basin, where we have acidophiles that are oxidizing iron, you're going to see a lot of red. And you're going to ask yourself, I wonder if that's a pigment or if it's iron.
So color is a good clue, but it also can be dangerous.
It can be dangerous. You got that, but it sets it up. It sets up the question in a nice way. It's like, oh, I know that iron could produce, if it's oxidized, could produce an orange or a red color. I also know that pigments of certain phototrophs in the right conditions could be maybe salmon or brown, and those colors overlap. So you got to be darn, you have to go into it one step further. And that would be measuring iron.
Right.
Is there even iron there? And that's a good key to know about.
Are there any features that are famous for iron in Norris?
Yes, definitely. Echinus.
Echinus, okay.
Echinus Geyser in Norris Geyser Basin has some of the most beautiful iron mats in the whole park. And at least that I was able to find. Now, I've seen a lot of iron mats because I studied those. And we looked at least 8 or 10 different types of iron oxide mats. And some of the most beautiful are in Echinus. We put up a little plaque there with National Science Foundation funds. Okay. So if you're out and about and you want to see this plaque, it's on the south side of Echinus Geyser. It's got an image, and of course you can see these beautiful terraced iron mats. Why are they terraced? It's because they form, basically it's like a chemical beaver dam.
Interesting.
Very nice, huh? You can look at it, and it'd be like a beaver, but it's a microbe. And instead of depositing logs to form a nice level pond, which is exactly what a beaver does, so it can live behind the pond, and the pond is perfectly level, the edge. The logs are perfectly level. Otherwise, it wouldn't be a pond. The microbes do exactly the same thing with iron oxide. They build up the lips of the iron oxide terraces to where it's like a perfectly level string of logs, but instead it's iron oxides. And the microbes live behind and on the edges of the dam.
Wow.
And it's a very fascinating situation. Terraces, and how do we recognize whether thermophiles live somewhere? Especially chemotrophs. If we don't see any color, a lot of times we have to look for the minerals they left behind, and that's what I'm getting at. In this case, the mineral has color, so it's red. But sometimes the minerals won't always have color.
Right, and that's a distinction, right, that you and I have talked about kind of outside the podcast. Is sometimes the color is the thermophile, right, or its pigments, and sometimes in this case, it's what the thermophile is doing.
Exactly. And the other main color that's useful to know off the top of your head is yellow.
Yellow.
For sulfur. Okay. And elemental sulfur, which is yellow as a solid phase, is very common and forms from the sulfide that we see coming out of the ground. We can smell it. So you'll often see some yellow. Now, what's interesting about the yellow is that there are thermophiles that can help catalyze the conversion of sulfide to the sulfur. So if you see yellow, there's a very good chance that there's some thermophiles there. Now, most of the time, when we see that high sulfur, not always, because it could happen in cold, it could be a cold seep also.
Okay.
But let's just say that if the temperature is higher than 74, then we're going to be certain that this yellow is due to sulfur. If the temperature is below 74 because of the photosynthetic limit, then we're not going to be certain that the yellow is from sulfur. So we're back to that situation where we really need to know the temperature.
Is there a spot in Yellowstone, like Aquinas, where you can go to see that we know it is sulfur?
Oh, yes, for sure. And Norris is another good place because we have acid conditions and we also have high temperature, which means that a lot of the yellow we see there is just from the chemical formation of elemental sulfur.
So all these beautiful colors kind of give you some clues, but it might take more investigation to determine exactly who's living there.
The measurements, chemical, and by measurements specifically, so we take the mystery out of that. First and foremost, it's a water sample. Okay. And we need to know what's in the water that's bathing the organisms. That's first and foremost. The next thing we need to know is the solid phases. If there's any minerals that are forming, that's going to be, a very, very important piece of information. Like, obviously, if we go to a place like Grand Prismatic and try to measure iron in the red zone, we're not going to find any iron minerals there. If we try to measure a pigment in the iron zone of a Norris geyser basin mat, we're not going to find any pigment. So those measurements come in extremely important to confirm. We can't just guess and say, oh, well, I think this, as you would imagine. We can't just use my tools to guess. We'd follow them up, of course. And that's why we have developed a really good sense of how temperature and pH, how they work in terms of sorting out, and of course, where organisms are. And then that comes with lots and lots of lab work where people have grown thermophiles under different conditions. It's not like everything we know about thermophiles only comes from our work in Yellowstone. Right. And that's important to remember as well, that many people, across the world have grown thermophiles under laboratory conditions to confirm a lot of what we're seeing in the field.
So we're talking about kind of how thermophiles, these thermophilic batteries, and how thermophiles use sulfur and iron, right, to drive their sources of energy. And I was curious, there are some that don't come with color, right?
Yes, there's some of the energy sources like methane and hydrogen. We're not going to be able to necessarily see that. Like if it's dissolved in the water, it's a dissolved gas, If an organism is using hydrogen, there's not going to be a color change that we can witness. If an organism is using methane as an energy source, there's no real color change that's going to necessarily be associated with that. So we're going to have to know that through chemical measurements. And that's the kind of data. If people wonder, if everyone out there wonders, how do we learn so much about various springs? It takes measurements of gases, the dissolved gases, to know that methane is extremely high, like Washburn Springs is one example. Extremely high methane, and we found several different organisms there that metabolize methane in ways that we would have never known.
Yeah, and we're going to jump into that the next episode with you.
Yeah, exactly. And so methane and hydrogen can be very difficult you would never know based on color, but we would know based on measurement of dissolved gases. And also, interestingly, sometimes we can use the genetics to go backwards and say, hey, if the organism has these genes, it must be doing this. And we can go back to the environment and then test that idea. We've done some of that, especially with methane.
I'm curious if someone's traveling around Yellowstone and after listening to this podcast wants to know more about who lives where. Are there any resources for that? To go look up hot springs? Is that data out there? Are there any guides to the thermal features?
Good question. You stumped me now. Yellowstone's got some good guides. I never read them now. No, just kidding. Thomas Brock, the father of, quote, thermophiles, that's a little bit of a misnomer, but Thomas Brock was very important in coming to Yellowstone early on to work on thermophiles. And I remember that the Yellowstone outreach programs, there's plenty of information on the description of thermophiles, and you yourself were involved in a film that long ago.
There's a film out there.
And that's a beautiful resource, by the way, is your film. I don't remember the title of it, but it was done about, let's say, a decade ago.
Yeah, a little less, I think, like 7 or 8 years. Yeah.
8 years?
Yeah, but it's on the Yellowstone website if people want to check it out.
Thank you. And that is, I have to say, that is an excellent resource. But you might remember the pamphlets, and you caught me off guard there because I don't know the pamphlet names. But there are several good ones that the park puts out.
Yeah, the park has a lot of resources. But I was curious if there's maybe a book or something. But I think Thomas Brock is a good resource.
Those are a little outdated now. That's the only thing I would say about going all the way back to Thomas Brock's little handbooks. And you say, well, why is it outdated? Well, it's outdated because the technology to prove and study, and then, first to study, and then to prove which organisms are there are just so much better now with genetic tools. And we'll talk about that more. But the genetic tools have allowed us definitive conclusions about who's there. And, moreover, physiologically, what allows them to be there. What are they doing? Are they oxidizing iron? Are they utilizing sunlight? We have the genes on all of that now. And that's something that Thomas Brock didn't have the benefit of having any access to. So he was doing it the hard way.
Yeah, I've always enjoyed traveling around the park and looking at the colors. I think people come and they snap a picture there and like, oh, look at those colors. But to me, having spent time with you and other scientists, it's a vast world you're looking at. And the colors give you clues, but they don't tell you everything. And that's interesting that they're these huge ecosystems of microbes out on the landscape.
If you're interested at all in having a tool that I believe is completely acceptable, is to own one of those temperature IR guns.
Yeah.
And the next tool that can be extremely useful is pH paper, but that would be against rules in Yellowstone's to be putting your hands and or anything into a spring. So we wouldn't recommend that, of course, obviously. But in general, I'm just going back to the two variables that are most important. So for the pH, you'll have to see if a sign and or a resource helps. And a lot of the signs do tell us, and because people have made measurements.
Yeah, and the park has good resources for pH of a lot of the springs that you can look up online.
Yeah, very good. And so, but wouldn't it be fun to be able to do experiments just as we're walking around? But that's not going to happen. We have to protect the resources. We have to. I think that you wanted to talk about this a little bit, even, and it gets me back to the importance of resource protection.
I'm curious. You've talked about gram prismatic a little bit and echinus, right? As someone who's done so much work on thermophiles, like give us a little tour of Bill's favorite spots to see different ecosystems. That could be a couple more, that could be a few more. I'm just curious, like, where would you go if someone hears this podcast and wants to see?
I think Norris Geyser Basin is a great place to start. Okay. I think the other place that I would contrast, I think you can see, first of all, a lot of diversity in Norris Geyser Basin. People say, or falsely, that it's only acidophiles that live in Norris. Of course not. There's some neutral sites there, but Norris is a place to go to see sulfur and iron, and outflow channels that have all three. There's some unique outflow channels in Norris Geyser Basin where you can see yellow, red, and green all in the same channel.
Wow.
And it's basically what is happening is sulfur is getting oxidized, then iron's getting oxidized, and then we're finally getting cool enough for phototrophy.
Wow.
And so it starts yellow, then changes, then there's a red zone where it's all iron oxide, and then you get into a green zone where it's these beautiful phototrophs. Turns out they're algae. Okay. They're not bacterial phototrophs. It's a very unique situation. It's a cyanidia. Is the name of the algae. It's one of the algae. There's a couple other types. These are thermoacidophilic algae, would be the proper name for them. But it forms a beautiful sort of rainbow right in front of your eyes. Now, so you asked me what was one of my favorite sites, Pinwheel. If you go over in Porcelain Basin,
okay,
and depending on conditions, if all the rivers are running right, you can see this yellow, red, and green right in front of your eyes, where it's a definitive line between the red and the green. And if you had a thermometer and could measure along that line, it would always be about 54 centigrade.
Wow.
Exactly. I mean, plus or minus half a degree. But the reason is that's as far as the acidophilic algae can tolerate temperature. It can't go any higher. It's a eukarya. So it's different. This is where it gets a little more complicated, because all of a sudden we got to think eukarya now. And so the green is only advancing to about 54 centigrade.
And if people want to get to Pinwheel, that's in Norris?
It's on the boardwalk in Porcelain Basin. And of course, keep in mind that the boardwalk in Norris will cover so much wonderful diversity. And then what we call the back basin loop. So there's two different loops, Porcelain Basin loop in Norris, and then the back basin loops, which will take you to the Echinus geyser, which I talked to you about.
Yep.
And I mentioned the iron terraces there are like no other that I've seen. They're just beautiful. They're basically iron oxide beaver dams. And you can walk over, it's a short half-mile walk over to Echinus. Then if you want to see phototrophic mats, some of the most beautiful phototrophic mats, I'd go over to Midway Geyser Basin, and even Upper Geyser Basin. I'd go over to Biscuit Basin and look at some of the color changes that occur in the outflow channels there. And most of those are phototrophic channels. So you get, what I'm trying to contrast for people there is a way to get both the chemical colors in your thinking, and then go over to a location where it's going to be more phototrophic colors. And then by getting both of those, you'll come away with, wow.
Yeah. And you might have to stick to Midway for the moment. I believe Biscuit is still closed because of the hydrothermal explosion. So we talked
that's right, Biscuit.
Michael Pulland in a different episode about that. And so they're still doing some monitoring there.
There are a lot of channels, though, right near there that are similar to the one I was thinking about with Biscuit. Yeah. But you're right. I'm glad you reminded me. That has blown up a couple times.
Yeah. Yeah, they just had it recently. Yeah, recently they just saw a minor hydrothermal explosion there on camera. So kind of expanding out, Bill, past viewing thermophiles and thinking about them in terms of these different parameters where they live, what kind of roles do they play in the ecosystem? You've already talked a little bit about building, right? Echinus builds these little iron dams. Are there any other examples, or what's their role in the whole Yellowstone ecosystem?
It's of course we learn in ecology that everything's connected. All life forms are connected, and of course that's true. But you have to be close to you have to be close to something to be impacted by it. Like an elk living up in, near Bozeman or something may not be impacted by all the hot water down there, if you see what I'm saying. Yeah. But so we have to start thinking, what sort of confines in terms of limits to the distances between spaces and what kinds of organisms would be impacted. But certainly one of the biggest interests is the formation of minerals, because that leaves a record in the environment, a geological record, like what we call stromatolites. And stromatolites is a word for fossils, essentially, microbial fossils, and their minerals that were important in, let's say, in the formation of early life. And so people love, not people, but scientists really like to get a hold of stromatolites to see if there might be clues about how life formed. So that's one of the more, you say, well, why, that's not a practical application. Well, no, this is a serious question about where do thermophiles come from and what do they mean for the origin of life and what do they mean for the evolution of all other life forms on the planet. So to me, that's a very practical question. It's very important. But yet, now, we can also talk industry, and there have been some very, very important industrial applications of thermophiles, but that gets to be maybe a little different question.
Yep, and it's something we have Dana Skorupa.
Yes, and resource protection becomes very important in that.
Yeah, let's talk about that. So why, that's a question I had here is, why is it important to preserve these areas? Like, why should people stay on the boardwalks and viewing areas, right?
Here's the thing. Some of the thermophiles that we've studied may only exist in really small pockets, like just literally a couple centimeters by a couple centimeters, maybe an inch. That's it. That's all you get. If you go in and accidentally sample the whole thing and then take it back to the lab and destroy it for your analysis, that could be it. And that is a very worrisome situation for the park, and rightly so, because some of the resources that we've studied, we don't know anything about. We could find genetics and mechanisms of how microbes might be doing things that could change literally energy production. It could change food production. These things are real. They could, and for example, of course, there's the wonderful story about high temperature polymerases, right, and the whole development of PCR. But those kinds of outcomes, which are not predictable, if we lose the resource entirely, and we just simply may not have known what we lost.
As someone, we're going to talk about your research in the next episode, but I was just curious, that's going to be very focused on the work you've done. How many open questions are there remaining? Like, how is there a vast amount of research still yet to be done?
Oh, yes. It's always funny how, after, let's say, 30 years, 35 years of doing science, you think that, oh, well, we know it all now. It's a good time to retire. That could be further, nothing could really be further from the truth. What's interesting is we've developed the tools necessary to have a much better appreciation of who's where and why. I think that now we're developing a better picture of how everybody interacts, and one area, for example, within that interactions question is how often organisms might be only doing part of a pathway and then an organism doing another part. So there's, hesitate to call it teamwork because that's a human term. It's not teamwork. It's just organisms doing parts of things that in the past we might have thought were compartmentalized. It's only one organism can do that. And now we're learning that no, 5 organisms participate in getting that done. So there's things that we're still learning about the microbial world, and the high temperature environments are perfect to study them because we don't have, in a lot of cases, these are simple enough environments that we may only have 2 or 3 organisms there, and we can get a sense of what they're doing and how they're interacting.
Yeah, and if those resources aren't protected, we
Yeah.
may lose the ability to learn from
Well,
those
indeed.
areas.
And some of them aren't very big. As far as, they don't occupy a large area, and researchers may not always know that. And especially, bison don't know it when they walk in the springs. And so Yellowstone can't. One time I was working down in Norris, for example, and it was, speaking of bison.
Yeah.
And bison jams. We were literally right in the middle of making a bunch of measurements in our spring. And I'd say about 60 bison started walking towards us. We just had to get out of the way. They walked right through what we called Beowulf Spring. They walked right through it. They basically destroyed it. For that, for that day, our sampling was done.
Wow.
We had to go home. We had to switch and go to the other springs we were working on. And it settled back down after a couple more weeks and reformed. But my point there is obviously we can't control what bison and elk do with geothermal springs. And you see a lot of interaction with the animals in the springs. In fact, it brings up another idea of a different study, but of how animals interact with springs. But the point being there is we do have to be aware as tourists and as researchers what kind of damage we might be causing.
So Bill, as we wrap up here, I'm curious just to ask you, as someone you said you've been doing this research for 40 years, right?
Yellowstone's more about 30, but
30.
Yeah.
So spent a lot of time in the park and done a lot of work with microbes and thermophiles. What about them fascinates you personally the most?
It's an interesting question because I've been really captivated by trying to come up with a theory that explains all of the diversity. And so when you ask me what's my favorite part of the diversity, it stumps me.
Okay.
A little bit. And that's a good thing because it forces me to think about, I think there's a few more things I would like to test, like ideas. And one of the ideas is, and we have not been able to test much of the mud pot environment. And the reason being, these are very hard systems to sample. Like when you look at boiling mud,
Yeah.
and you those ones that, and I think if we've been in Yellowstone, we can hear them in the background. They're sort of bloop. Then another minute later, bloop.
Yep.
And those environments have not been studied well. And I'd be fascinated to get into some mud pot work. Now, they may be simpler than we expect, perhaps, or because there's been little work, we don't know what to expect necessarily. But the environments may not be that complex.
Okay.
Especially if it's all one mineral, like kaolinite, like a clay mineral. And it's a lot of times these are low pH environments, and so basically you're getting a lot of dissolution of the mud, and it's they're very hard to sample because there's not much. The water content is easily extracted out of there to make measurements of. So I'd say environments that we haven't looked at, mud pots, bottom of the lake. That's another area that we're going to talk about later. But that's an area that we have not been able to investigate because of the logistics of getting there. So that's a big interest of mine. And the discovery of new life forms, or of new organisms that nobody's known anything about. Now, we've made good progress there, and I don't know who knows how many are left to discover. It's amazing the rate. During the genomic revolution, how many new let's say, divisions of organisms were actually discovered, but it's been many.
All right, Bill. So I kind of want to wrap up this episode with one question. I'm just curious. We've talked a lot of chemistry, a lot of detail today. Hopefully people have learned a lot about thermophiles. But if they take away one thing from this episode, someone who's learning about them for the first time or is a park visitor, what do you as a thermophilic researcher hope people take away about thermophiles?
Can it be more than one thing?
Yeah, of course it can be more than one thing.
I want people to take away as a couple really important things. First thing I want people to take away is thermophiles were probably their relatives were the origins of life. That's it. That's the first thing you got to take away. It's like, these are our ancestors. Now, and of course, then you can think about all the evolution that had to take place. But scientists have evidence that life started in high temperature environments. So that's very important to take away. The next thing to take away is, these are distributed in different environments just based on a couple simple variables that we all know, temperature, pH, then different chemistry. That's it. If you can take away those two things, you can drive through the park knowing everything that I would know to start making guesses and hypotheses about what might be living there. So I think that's enough.
Great.
Those two things.
Well, thank you, Bill. I think that's a good place to wrap up this podcast. I want to thank you so much for joining us today.
Thank you, Hugo, for having me. It's, like always, wonderful to talk about Yellowstone.
Thanks for coming. It's always great to chat with you. And for our listeners, be sure to follow along as we continue to explore the hot side of Yellowstone in future episodes. Thanks for listening, everyone, and we'll catch you in the next episode. Bison Jam is a production of Hugo Sindelar Media, LLC. I am the director and producer, and Ashley Bao is our editor and story consultant. This episode was filmed by Braden Meyer. Our guest was Dr. Bill Inskeep, Professor Emeritus in the Department of Land Resources and Environmental Sciences at Montana State University. Additional support for this podcast also comes from Montana State University. Thank you again for listening, and be sure to check out future episodes wherever you find your favorite podcasts. See you soon.